Click for IB Biology. Here we're looking at the functioning, the structure and function of ribosomes. Their combination to polysomes will be considered briefly, and their association with endoplasmic reticulum in what is called rough endoplasmic reticulum. So we'll take a look at that organelle as well. There's a blank PDF of this presentation down in the description, which you can annotate with your own notes. And then, of course, there's a quiz with 10 questions and answers for you to check your progress. So let's begin with the idea of ribosomes, which are free within the cytoplasm. Now, when we use the phrase free, as we've done here, we mean that the ribosome is operating by itself. It's not associated or attached to the outside of something like the endoplasmic reticulum. And where this is the case, the function of the ribosome is to produce proteins for intracellular use, so within the cell itself. Analysis of the structure of the ribosome shows us that it's actually composed of two parts. And here I've labeled these as the small subunit and of the large subunit. So the small subunit's function is to lock onto the copy of the gene called messenger RNA and to create the sites called codon, anticodon binding sites, which will allow for the synthesis of the amino acid sequence and the polypeptide. So this is where we're actually seeing the genetic code being turned into the actual protein, which of course is a process called translation that we'll look at in more detail in other videos. So back to the structure of the ribosome. It's in two parts. In eukaryotic organisms, then the type of ribosome is what's called an ATS, so it's a large ribosomes, Feldberg units. The diameter across, the distance across a ribosome like this is round about 35 to 40 microns. Sometimes it depends. Some are smaller and some are larger. And so you can see the messenger RNAs here, the tRNAs here like this. And today, with modern electron microscopy, we're now able to image these molecules using cryo-electron micrographs like this. And this is an image here of just such a ribosome. Within the ribosome, there are actually some proteins as well. I believe in eukaryotic ribosomes, there's about 49 different proteins. Prokaryotic ribosomes are slightly smaller, which is one of the distinctions between pro and eukaryotic cells. So the overall function of a ribosome is the catalysis of the peptide bonds, joining amino acids together in order to create polypeptide chains. And this happens within the cytoplasm. Now, interestingly, this micrograph here will show us another feature, which is the messenger RNA is along my pen line here, and there are actually multiple ribosomes here. And the lines which are out here, these are our polypeptide chains up here. And so we've got many ribosomes attached to one messenger RNA, and that is called a polysome. And the purpose of that is it greatly increases the efficiency of making of the proteins. So single ribosomes, intracellular proteins, proteins to be used within the cell. Now contrast that with the ribosomes which are attached to the endoplasmic reticulum, and when this happens, the reticulum, which we can see as these lines in this micrograph here, gives this grainy appearance of dots around the outside of this tubular system. So the endoplasmic reticulum is a tubular system, and we can see these grain-like structures here. Those are the ribosomes. So in combination, this is called rough endoplasmic reticulum. And what we know is that where we have rough endoplasmic reticulum, the proteins which are being made on this are destined to be used outside of the cell. So as an example, if you were making the protein insulin in a pancreatic cell, then the amino acids would be joined together on the ribosome, but it would happen on rough endoplasmic reticulum ribosomes. And then, so they're isolated. You can see here, these are the ribosomes. And then the protein which has been manufactured is then introduced inside the endoplasmic reticulum, into the lumen of the reticulum. These tubular systems are about 20 nanometers in diameter. So now we're down to a much smaller scale measurements of nanometers rather than micrometers. So 20 nanometer diameter from here to here. And the protein will go in here, where it will probably be modified prior to being released from the cell. And it's at that point that the rough endoplasmic reticulum, since it's going to send its proteins to the outside of the cell, is always found in conjunction with the Golgi apparatus. And that's an organelle which we'll discuss in our next video. So the tubular system of the endoplasmic reticulum is for the production of proteins, their modification, packaging, ready for secretion from the cell, perhaps into the blood, perhaps into the extracellular fluid. The links to those videos, to those other organelles rather, are now occurring in front of us. There's the link to the channel and, of course, to the playlist.